Hello everyone, our group will be presenting on an experiment involving cytokinin. This is the list of our group members, and I will be giving a little bit of introduction on cytokinin. Cytokinin is a hormone which is a plant-specific chemical messenger that plays a central role in the regulation of the plant cell cycle and numerous developmental processes. Several cytokinins occur naturally in plants. They have an adenine base and a 5-carbon isopentyl side chain. Among these, zeatin, specifically transzeatin, is the most abundant. They move upward in the xylem, which is the woody tissue part of a plant, and pass into the leaves and fruits, where they are required for normal growth and cell differentiation.
Now, I would like to share four main functions of cytokinin. Firstly, cytokinin promotes cell division. It helps in growth and development of the plant. Secondly, it delays leaf senescence. Cytokinin aids in preventing the leaf from yellowing. Cytokinin also can be used as light responses. It regulates the photosynthetic pigments and protein synthesis on the irradiated leaf. Lastly, it can also be used as nutrient mobilization. It influences the nutrients in plant to move from leaf to the other parts of the plant.
Now I will talk about the methodology of our experiment. Firstly, a plant was obtained and about 5 internodes of the stem consisting of leaves were counted. The stem consisting of leaves were being cut and it was cut slightly tilted. Then, step 1 and step 2 were repeated for 9 more times so that the total of stems obtained were 10. After that, 10 flasks was prepared. 5 flasks is for cytokinin hormone and another 5 flasks is for water. The stem were being placed into all 10 flasks with the stem being immersed in the solution. Then, several leaves surrounding the stems of the plants were removed and the flasks were covered with plastic so that the water and the hormone will not be evaporated into the air. After that, the flasks were placed near the window, leaving the surface of the leaf being exposed to the sunlight. And lastly, it were left for several weeks before being observed and recorded.
After a few weeks for the plant grow in water, there are a tiny shoot grow from the lateral bark, which shown in the video below. For the plant grow in cytokinin, we observe that there are a few branches growing from the lateral bark at the end of the experiment, and this is shown in the video below.
From the result, we could see new shoots already grown from lateral buds. For plants without cytokinin, we can observe that only a tiny shoot has developed. While for the plants with cytokinin, there are many branches developed and the development of fruits is very well. Cytokinins are essential to plant hormones. The main function of cytokinin is to promote cytokinesis or cell divisions. Thus, by stimulating cell division, they regulate shoot meristem size, leaf primordial number, and leaf and shoot growth. Cytokinin deficiency causes slow growth of the shoot and morphological changes such as dwarfism and smaller leaves.
Must be discarded totally. Although cytokinin does not have bad effect on humans or animals, we should not let cytokinin contaminate streams, ponds, and lakes, as water life may be endangered. The work table must be always organized and clean. And to ensure positive results, the suction use must be measured accurately, and the outcomes of the result must be observed and recorded.
To summarize, in this experiment, our qualitative data obtained. However, for our quantitative data, we could not obtain it. This experiment did not achieve our objective, since only qualitative data being received. However, by our qualitative data, cytokinin acts as hormone in the plant. It is essential for stimulating the cell division and play important role in the plant. Thus, the reason why this experiment did not achieve the objective. Limitation of cytokinin and members of the group separated from each other has also led to this result.